So this topic is about human engineering and scientific aspects of disease transmission in natural and built environments. And our first speaker is Claudia Gunsch. She is a professor of civil and environmental engineering and holds secondary appointments at the Nicholas School of the Environment and the Department of Biomedical Engineering. Gunsch currently serves as the director of PreMiEr, the National Science Foundation Engineering Research Center for Precision Microbiome Engineering, which is a joint venture between Duke University, North Carolina, A&T State University, North Carolina State University, University of North Carolina, Chapel Hill, and the University of North Carolina, Charlotte. She also serves as an associate director for the Duke Microbiome Center. Welcome.
Great, thank you. Good morning, everybody. It's a pleasure to be here today. As Mark mentioned, I'm a professor at Duke, and I have the pleasure of serving as the director for the NSF ERC for Precision Microbiome Engineering, or PreMiEr. So today I wanted to just provide a brief overview of what the center is all about, and then zero in on approaches to microbiome engineering that are specific to the built environment. So as was alluded to in the introduction, we are a engineering research center that focuses on the built environment and microbiome engineering in that context. We are a collaboration of 5 different institutions across North Carolina. The headquarter institution is Duke University, and we have the 4 partner institutions. North Carolina A&T, where Dr. Joe Graves, on the second line, I don't know if I can actually point to it, but Dr. Joe Graves is the PI at North Carolina A&T. His expertise is in evolutionary biology. At North Carolina State, Dr. Jennifer Kuzma is the director, and her focus is on societal and ethical implications. She has done a lot of work in genetic modification and genetic engineering in general, particularly as related to food. And so feeding off of the conversation earlier, thinking about public attitudes and acceptance of some of the technologies that will be coming out is an important part of the work that we do. University of North Carolina, Chapel Hill, where Dr. Jill Stewart leads the effort. She's also the deputy director for the center, and her work, she, as many of you know, is really focused in microbial ecology. And then finally, University of North Carolina at Charlotte, and Dr. Anthony Fodor is the lead at that institution. His background is in bioinformatics. Much of his work has been focused on developing pipelines related to human health, and now thinking about how that work can tie over to the built environment in general. But beyond the 5 of us, we bring together a diverse group of engineers, microbiologists, physicians, bioinformaticians, and you can go down the line. I'll skip to entrepreneurs and social scientists to really think about what is the impact of the work and making sure that the work we do is spun out of the laboratories. We've actually grown. This is a picture that was, or picture summarizing all of the participants at the end of year 1. However, we have had a few new additions since then, so stay tuned. So at PreMiEr, really what we are focused on is understanding the microbiomes of the built environment and how they affect human microbiomes and human health, something that's obviously of interest considering the workshop that we are all attending. Our work is focused on 3 main prongs. First is the measurement, to know who is there and what they are doing, and also thinking about what are signatures that are indicative of presence or absence of particular organisms. The second prong is modeling, thinking about developing predictive models that incorporate that data. And the third one is modification. So thinking about what are strategies that we can use to manipulate and engineer that environment. So the big questions that drive our work is, as I mentioned, how does the built environment lead to the transfer and colonization of particular microbes? And secondly, how can we actually manipulate the built environment to engineer solutions for reversing risks? And a lot of the motivation for our work is thinking about the increase in immunomodulated diseases and the knowledge that the microbiome plays an important role in progression of those diseases, as well as response to particular medical treatment. And so thinking about how those individuals may be acquiring microbiomes through exchanges from others, as well as their interactions with the built environment. The work that we do is centered on our test beds, and I will talk about at the very end of today's presentation the test bed models that we are using and how we hope to have these as a service to the broader community so that we can put our efforts together. But we are working in these test beds and obtaining the data for the different research thrusts, developing the models, as well as testing our modification approaches. All of our work sits in a common data analytics core. We know that the power of our work will only come from having this common infrastructure. We also know that within the systems that we are working in low biomass environments. And we know from a recent paper that came out in Nature, thinking about the fetal microbiome, that not taking into consideration some of the challenges that are associated with low biomass environments can actually lead to misinformation or misunderstanding of the importance of particular microbes. And so we're working on developing approaches and pipelines that enable us to work in those spaces. And then finally, the work that we do is anchored in our SEI core, the societal and ethical implications, tying with the work that we talked about in the first session, thinking about the implications of the work that we're doing and making sure that there's public awareness and also public acceptance of the approaches that we choose to pursue. So you are most likely very familiar with this report from the National Academies, the microbiomes of the built environment, that really set the research agenda for work in this particular space. We know that there are these links between occupant factors, building design, ventilation, and so on and so forth. But what is important to note is that as researchers and engineers, we tend to gravitate towards thinking about impactful studies that give us very high power. And by what I mean by that is that we tend to work in systems that may minimize environmental complexity so that we can really start understanding how all of these pieces fit together. When in reality, the real world is at the opposite end of the spectrum in terms of environmental complexity. And this is really what drove our selection of the test beds within our center, so that we can really start thinking about testing hypotheses, developing hypotheses, and going back and forth between these different levels of complexity that represent the real world. So general approaches for engineering microbiome really falls in three different buckets. The first is the physical features of the particular built environment. What are the materials? What are the coatings? Even we know that different food that people ingest will be associated with particular microbiome signatures in the built environment. The second one is thinking about just operational. How are we operating the systems, the plumbing system? We know that there are microbes that come from the premise plumbing, the HVAC system, how the use of filters and just the general operation of those systems will affect microbiome signatures. And then finally, and this is where a lot of the novelty comes in, is how do we think about biological interventions of the microbiome, similar to those that may be used in medical or agricultural fields, the addition of encapsulated microbes, for instance, those that we know may be beneficial. Can we start thinking about horizontal gene transfer of particular plasmids to start modeling antibiotic resistance or even preventing antibiotic resistance, bacteriophage applications? Can we start thinking about signals and quorum sensing or even CRISPR technologies? So as I mentioned, this modification and microbiome engineering has really traditionally focused on medical and agricultural applications. And a lot of the work that we're doing is starting to think about those particular applications, but in a different context. And so this is a figure from a review paper that really focuses on some of the general approaches, antibiotics, just killing everything that's there, probiotics, can we start thinking about replenishing those microbes, fecal microbial transplants is another approach, and now moving to very targeted applications. And so this is where we come in and thinking about what are delivery tools that lead to targeted colonization as well as expression in the built environment. So not only are we thinking about what are microbes that we can incorporate or subtract from that environment, we want to develop tunable expression systems that may be able to focus on just particular features of those microbes. So in terms of delivery vehicles, the ones that we are looking at are encapsulated microbes. This is an approach that has been used, and even in my laboratory, we're using it in the context of bioremediation. Bacteriophage, so thinking about the application and delivery of particular phage that can target particular organisms of interest, and then plasmids, thinking about a beneficial transfer through horizontal gene transfer of plasmids of interest. So our work in microcapsule delivery has focused or is focused on premise plumbing systems. We are developing a variety of biopolymeric microcapsules that can be delivered to premise plumbing. We're focused on premise plumbing because we know that there have been a number of instances of re-aerosolization of pathogenic organisms in those systems, particularly in hospital environments. And this focus is thinking about, can we introduce known organisms that will prevent the colonization of those trouble pathogenic microbes. Another aspect of our work is focused on modeling the introduction of particular phage. This work is led by Professor Mark Wisner, who is an expert in understanding these interactions for nanoparticles and thinking about models of heteroaggregation. And if we can understand that, we can start applying these particular theories and models to phage bacterial interactions and really starting to understand which phage may be introduced for particular, to kill off particular bacteria. As I mentioned, we're also interested in functional modulation, and this work is led by Dr. Nathan Crook and Dr. Kevin Garcia at North Carolina State University, and here focused on conjugation. Can we promote or prevent particular conjugation efforts to eliminate targets such as antibiotic resistance? And I'll talk about that in my next slide. And then also developing systems for non-model organisms and recognizing that the work that we do is in non-model organisms. So understanding how we can manipulate and develop these fine-tunable mechanisms for the systems of interest. For conjugation, a lot of the work that we are doing is being led by Dr. Ling-Chong Yu at Duke University, and his expertise is really bridges the computational aspects of modeling transfer of particular plasmids by horizontal gene transfer in the context of antibiotic resistance, as well as the actual work in the lab to be able to test those models. The interest of ours for this particular work is thinking about what are the trade-offs between vertical and horizontal gene transfer that are linked to plasmid fitness in a broader context. And if we can get to the bottom of that and really understanding that in a complex community, we can start altering that particular microbial community to prevent the transfer of antibiotic resistance. With that in mind, we can now start thinking about not only bioaugmentation and really thinking about how can we modify that community structure if we can understand how introducing particular microbes can lead to a modification of the broader community, but we can even take those same concepts and start applying them to environmental sensing and actuation in those complex communities. So I'm getting close to the end of my talk, but I wanted to just introduce our test beds. And going back to what I mentioned initially, we are collecting our data in and testing and developing our hypotheses in this range of test beds, ranging all the way from very simple systems where we have high levels of control in our environmental air chambers in Dr. Turpin's lab at the University of North Carolina, Chapel Hill, all the way to the very complex systems, the work that Dr. Joe Brown is leading in Bolivia, thinking about how can we look at built environment microbiomes in a context where there is no control. There's no HVAC systems in these particular homes. And so that's the opposite end of the spectrum. But I wanted to draw your attention to the test bed in the middle. This is the PreMiEr home test bed. And so this is a home on the Duke University campus. It's about 4,000 square feet. And our vision for this is to really now start thinking about a model that is representative. And you can think of this similar to the NIST test bed models that are really focused on the ventilation and some of the chemical characterizations of airborne and aerosol transport, but now thinking about it in a microbial context. We have a number of rooms that are present and hoping that this will be of benefit to the community. So my watch is buzzing, which means I'm at time. But with that, I will thank you. I will point you to our website as well as our other presence. And we hope to stay connected with you. And I look forward to the Q&A section. Thank you.
Thank you, Claudia. So the next presenter is Dr. Yun Shen. Dr. Shen joined the Department of Civil and Environmental Engineering at George Washington University on September 1st, 2022. Her research is tackling the challenges on solutions to health environment nexus. In particular, she is investigating the transmission and control of environmental pathogens, including transfer, persistence, infectivity, and virulence, and inactivation and removal of pathogens in a variety of environments, including wastewater, drinking water, soil, and indoor air. She is also investigating the interactions between pathogens and other emerging contaminants, such as microplastics, in the environment. Her research is supported by NSF, EPA, USDA, and NIH. Prior to joining GWU, Dr. Shen was an assistant professor in chemical and environmental engineering at University of California, Riverside from 2020 to 2022. Thank you, Dr. Shen.
Thank you. Thank you for the canned introduction and good morning, everyone. And today I will talk about the viral clusters in wastewater, which is an overlooked form of viruses. So, each year in the world, we have millions of people died from infectious disease, and especially from the respiratory infections and diarrhea disease. And the loss of the respiratory infections and the diarrhea disease are actually caused by the virus transmission through air, water, and food. And for respiratory infections, viral infections actually contributed to over 60% of the infections in children. And for the diarrhea disease, the rotavirus itself caused like more than 0.5 million of deaths in children. So the viruses do raise human health concerns in the environment. Wastewater is an important reservoir of many of these viruses. So the viruses can be present in human waste and then reach to the wastewater treatment plants. And in this figure, so it is actually from a review paper, and it shows a summary of like the concentrations range for different viruses. So we can see that like for some viruses, such as like this one, the human adenovirus, even after the treatment in the effluent, as shown in this green bar, the concentration can be as high as 10 to the 9th gene copies per liter. And those viruses in the effluent, they can then finally reach to humans through the potable water reuse, through the agriculture irrigation, and also the recreation water. So therefore, understanding the presence and transmission of viruses in wastewater is important. So in particular, we want to understand the transmission of viral clusters in the wastewater. In the past, we treat viruses as free and single particles. However, the viruses, they can actually aggregate together. They can adhere to like other particles, such as the clay, the plastic, the bacteria, and they can also be cloaked in the vesicles released from the host cell. And a few studies found that those viral clusters, they can be prevalent, they can be more infectious, and they are actually more resistant to disinfection. So in our research group, my student Yihan Wang is actually studying the adhesion of viruses on nano and microplastics and their infection and disinfection. And in this figure, Yihan studied the adhesion efficiencies of a coronavirus strain on 3 different types of plastic particles and also the silicon particles. So we can see that actually a large amount of viruses can adhere to those particles up to the efficiency can be up to 30%. So that implied that in the environment, actually lots of viruses, they are banded with the particles. And another student in my group, Xiaonan Tang, and she's studying the influence of bacteria on the infectivity and the resistance of viruses. And later today, she will have a poster and welcome to stop by. So from her preliminary data, we found that some bacteria, they can largely absorb the viruses, and they can enhance the infectivity of some viruses. So those are some ongoing studies in my group for the particle-associated viruses. And in the rest of this presentation, I will talk about the vesicle-cloaked viral clusters or the viral vesicles in the wastewater. So the viral vesicle was just discovered in recent year, but it has been recognized as a major form for some viruses. And when the viruses propagate in and released from the host cells, it's possible that multiple viruses can be cloaked in a vesicle. And the vesicle has a lipid membrane and the size of like from 50 to several hundred nanometers. And those are some like TM image show like what do the viral vesicle look like. And previous studies found that a large portion of viruses can associate with vesicles. For example, the rotavirus was found to like the vesicle can contribute nearly half of the total virus in the stool samples. And also those viral vesicles, they are more infectious than free viruses, and they are more resistant than free viruses like under the UV exposure. So however, like although we know the biological and health significance of those viral vesicles, no one have ever isolated or detected the viral vesicles from any of the environmental samples. So we are going to fill the research gaps. So here are the research questions we want to study from our study. First, we want to know if the viral vesicles present in the wastewater. And secondly, we want to know like what kind of viruses can associate with vesicles in wastewater. And the last, we want to know like will those viral vesicles challenge our current wastewater treatment strategies? So we are collaborating with Professors Tian Ao, John Rose, and Damon Shuai's groups in this study. And also this study is relevant to 2 of our workshop topics, the engineering approaches and also the monitoring of disease agents. So to answer those research questions, we collected raw wastewater from a municipal wastewater treatment plant and a hospital. And then we separated the wastewater and the sludge, and we utilized the antibody-coated magnetic beads to isolate the vesicles from those samples. And after that, we extracted RNA and DNA from those samples to quantify the norovirus from these samples, because norovirus is the most common foodborne pathogen. And we also conducted metagenomic analysis to identify the types of viruses associated with the vesicles. So did we successfully isolate any viral vesicles? So after the isolation, we checked the samples by TEM, and we found the vesicles that containing the viruses by TEM. And then we also confirmed the size of the isolated vesicles by using the nanosite. So the size is in the range of what we expected for the vesicles. And then we want to know, like, can we detect any norovirus in the isolated vesicles? So we conducted a qPCR to quantify norovirus. And in this figure, we have the concentration of norovirus G1 in the hospital wastewater. So the x-axis is a sampling date and the y-axis is a concentration. So we can see that for both so in this figure, the solid line represents the total norovirus G1, and the dashed line represent the viruses associated with vesicles. And we can see that for both total and vesicle-associated viruses, like the highest concentration observed in winter time and then decreased. And the ratio of the vesicle associated to the total norovirus G1 is about 43%. And we also did similar analysis for the sludge sample, and we observed like a similar seasonal change. So the highest concentration is in the winter, and we also observed a significant amount of vesicle-associated norovirus G1. So we also tested the concentration of norovirus G2. So we tested both norovirus G1 and the G2 concentration in sludge and in wastewater from the hospital and the municipal samples, and we found like similar seasonal change, and the average ratios of vesicle to total norovirus ranged from 17 to 45%. And we also compared the ratio of vesicle associated to total virus for both norovirus G1 and the G2. So those are the two most common genotype that are causing the gastroenteritis. And in this figure, the x-axis is sampling type, y-axis is the ratio of vesicle over total, and the red color represents genotype G2, blue color represents G1. We can see that the ratio for the norovirus type G2 is always higher than G1. And previous studies also found that norovirus G2 is more prevalent in the environment. It's more resistant to disinfectants, and also the majority of the norovirus outbreak is actually caused by norovirus G2. So these findings on like G2 might be attributed to their preference to associate with vesicles, because vesicles can make them to be more infectious, and they can protect the viruses from like environmental stresses. So to summarize the first research question, so we found a significant amount of viruses that can associate with vesicles, and also norovirus G2 tended to associate more with vesicles than norovirus G1. And then we can take a look at the second research question. So will the viruses be, what kind of viruses can associate with vesicles in wastewater? So to answer this question, we applied the metagenomic analysis. So here I only have time to show the analysis results for the RNA viruses. So as shown in this figure. In this figure, so we have like 4 different types of samples from hospital and municipal wastewater or sludge. And then in the left side, we have the total virus. In the right side, we have the vesicle-associated viruses, and like a different row actually represent a different type of viruses. So we can see that the 2 viral communities for total and vesicle associated are actually different, very different. And we applied the principal component analysis to prove that these 2 viral communities are distinct from each other. And that means the vesicles, they can select some viruses. And especially, so for some viruses were only found in vesicle samples, but they are not found in the total viral communities. That means vesicles can enrich those viruses. And in addition, we also found several human viruses that can associate with vesicles. So that include the norovirus, rotavirus, the astrovirus, the saparovirus. So those are all very commonly found the viruses to cause the gastroenteritis. And also there are actually lots of the plant viruses. So we know that the treated wastewater are actually frequently reused for agriculture irrigation. So those viruses may cause some concerns on the agriculture production and the ecosystem health. So from the metagenomics, we identified some like human viral pathogens associated with vesicles. So the last question is, will those viral vesicles challenge our wastewater treatment strategies? So we don't have the answer now, but we are actually testing the removal efficiencies of those viral vesicles in the real wastewater treatment processes. And we will have more information at that time. So like after understanding like the abundance of viral vesicles in wastewater and the types of viruses associated with vesicles in wastewater, that actually advanced our knowledge on the risk of viral vesicles in wastewater. So we need to be more cautious on like those viruses that can associate with vesicles, because they can be more infectious and they can cause maybe higher health risks in the wastewater. And with that, I would like to thank all my collaborators and the funding agencies. And also I'm ready to take the questions in the Q&A session. Thank you.
Thank you, Dr. Shen. So we're gonna skip over to the Q&A session.
To my question to Dr. Shen, I was just wondering, why is the reason that the virus concentration decrease in summer, like it's higher in winter and lower in summer? Is there any particular reason for that?
Sorry, can you repeat your questions? Your question is about the
concentration of virus in water, that it's high in winter and low in summer. Is there any particular reason for that?
So that actually also agrees with the trend of the norovirus outbreak. So it's like always higher in like the winter time. So that might be because like in the winter time, like the temperature is low And also like we have like a dry environment, and it's easy for the viruses to like to survive and transmit, and also like maybe it's also related with the like a human immune system in the winter time. So it's more like susceptible to those viral infections.
Understood. Thank you.
Okay, thank you.
Thank you for your question. Go right ahead.
Thank you. My question is for Dr. Shen. Really nice talk. So for those of us who work in risk assessment, aggregation of norovirus ends up being very important for the modeling efforts, and it seems that it's a lot more complex than maybe folks have given it credit for. Do you have any recommendations for folks doing risk assessment? Does it need to consider like specific chemistry of the matrix or location or timing? Maybe you can speak to that a little bit.
Yeah, thank you for your question. It's a very great question. So so far, I don't think there are lots of like informations on the disinfection kinetics for the like for those viral clusters. So we are actually doing some at this moment, and so without like those experimental information, it's actually hard to predict. I think once we have more information available for like the disinfection kinetics of those viral clusters, it will be very helpful for like the risk assessment. Thank you.
Okay, great. So I have a question for Claudia. So I'm just wondering, because when we talk about built environment microbiome, it's an open system that is always changing depending on the environment. So, and you have talked about a couple of different approaches for the microbiome engineering approaches, and I'm just wondering how sustainable all these approaches are. For example, if you do functional modulation, or you do a encapsulated microbiome inoculation, how long this beneficial effect will last?
Thank you for the question. So that's what we're trying to get to the bottom of. We don't really understand yet even how long particular microbes stay in a particular environment, let alone what these alterations will do. I would say that the best example that I can provide is we know, for instance, in a hospital room, there's been a number of instances where individuals in a hospital rooms, you can actually follow the progression of a disease being an infection coming from a P-trap. And so thinking about, and I should say, and we also know that we can model when somebody comes in or a new patient comes into a hospital room, within a couple of hours, the microbiome in that space has been completely turned over. So we can start thinking in those particular environments that there may be interventions that we can perform in the P-trap to alter that particular transmission. But you're entirely right. When we go to the very complex systems, such as the Bolivian home, where there is no control of that, then here we're really looking at how are things being transferred between the ambient, the outdoor environment, and the individuals in the home. And that becomes a really complex question. And there's too many variables, unfortunately, for us to be able to answer it in that context. So we're, starting from a situation that is much more controllable to see if those particular models then carry over. Thank you.
Are there any other questions? Yes, go ahead. Oh.
Okay, hi, I have a comment and then a question. Comment, I really appreciate Dr. Shen bringing up the virus, the vesicle-coated viruses. I think that one thing that we need to figure out more as a field, Mamadou, is like how pathogens actually exist in the environment. What state are they in? What are they expressing? Are they coded? Are they protected? How do they behave differently in its viruses and bacteria? And then we're not talking as much about protozoa and helminths right now, but I don't want to forget about them. But I think definitely for the viruses and bacteria, it's really important for us to understand in terms of figuring out controls, how are they in the environment? Because so much work has been done with with lab grown, lab strain organisms. So thank you for that. And all that work that you're doing is like super cool. Yeah, it's really nice to see. And then I wanted to ask, in terms of the microbiome work, you kind of alluded to this, but I was very fortunate to hear Amy Mathers talk about work that they were doing in, I think, UVA, where in the hospital, they realized that there was a lot of like antibiotic resistant hospital acquired infections in their hospital. And what they, after a lot of searching for the source, realized it was the drain. So like it was actually living in the drain. And when you turn the water on, it splashes out of the drain, goes everywhere. And then it's a huge problem. And so I was wondering if you guys are also looking at materials. So I think that's a really interesting question about like, okay, if you seed a microbiome, how long does that last? But are there ways we can enrich or prevent based on materials? So thanks.
Wait for that. No, it's green, so it's good, I think. Yeah, so we are thinking about going back to that approach. There's really the three facets. One is, how do we actually build and architect and those particular spaces? Then how do we operate them? And then there's this biological control element that comes into it. So you're entirely right. That there is that element. And seeding into what you just mentioned, there's also, if you think about the sink architecture, there is actually a link with socioeconomic status also. If you think about the particular faucets and think about the splash zone, thinking about if you go into a newly built house now that is in a more affluent neighborhood, you're going to see deeper sinks. You're also going to see a faucet where that splash doesn't occur more or isn't as big. But if you go to a low income housing, what you end up is with these sinks that are actually pretty shallow, and you start looking at the actual faucet itself and how the water comes out, and that's going to impact, one, how the splash zone occurs, but then also what is being what's growing in the biofilm that is actually developing in your P trap, which is what the drain, the work that they've done at the Virginia points to. So there's a lot of work just thinking about that particular piece. So I'll just put a plug. This is something that we're really interested in doing, is just a citizen science project. So we currently have students in our first year design course that are helping us develop kits that we can send out where people could literally just put these inside of their sinks, so that we can start understanding how individuals will lead to the development of particular biofilms and P-trap microbiomes, so that we can start looking at that. But yes, you're entirely right. We have to look at the whole thing. I mean, if you're looking at copper pipes, it's going to be completely different than if you have PVC. And you were talking about microplastics and how that affects. So we can't be considering all of these things in isolation. So yeah, thank you for your question.
Question I'll ask. So just two questions for both. I mean, so the work you're doing in clock, basically, so can you have instance where many different types of viruses, when you have aggregation, it does not have to have uniform, viral composition. It could be a mix. Like, can these things deliver really mixture of really pathogenic viruses? That would be scary.
Yes, it's green. Yes, I agree that it's always a mixture of like all those different types of viruses and in different forms, like free living pathogens like bending into other particles, the aggregates of pathogens. Yes, so it is like a very complex like environment and a system to study. And yeah, I think so for now, like we want to study that from like a more like a harshable like viral forms. And once we understand that, and we can go into like a more details on like what will be like the treatment strategies for like all those mixtures of like viruses or bacteria with different types as a whole. And yeah, so I think we now, since we noticed that those pathogens can have like those different forms, it's a good start, and we started to look at them, and later we can like have better strategies to control them.
So question for Claudia. I mean, there is a lot of work recent in, the gut microbiome. There are many things like the Engineer Biology Consortium, NSF funded a workshop on microbiome, and they think of microbiome as like, let's say, three things: functional diversity, spatiotemporal control, and distributed metabolism. Can you think in the built environment? Can you look at, spatiotemporal control? Can you comment on diversity?
Yeah, just to add to the previous comment that you made to Dr. Shen, I was going to mention there's a huge need for just surrogates for us to be able to study, because a lot of these pathogens and viruses, we can't study them in these complex environments. And so I think there's a huge research gap in terms of actually having either synthetic microbes or viruses that we can actually study in these complex environments. So that's one plug I'll put in there. I think there's a huge research gap. To your point, yes, all of those, and I think that's why we need models to be able to look at that. Right now, I would say we do not understand how to even model, how to measure, what is the density of measurements that we need. And so this is where our test home, we're hoping that we can really map it at that level to provide this as a service, not only for what we're doing in our center, but also just to the broader community, for us to start thinking about those questions. And then that feeds into the third part with the metabolites. So right now there's a huge gap in terms of even a library of microbes that are relevant to the built environment. The reason that we've made so many advances in microbiome engineering of the gut is because now we can actually go in and look, and with pretty high degree of certainty, we know who is there. So we can start interrogating and developing strategies that target particular metabolites. We are nowhere near that in the built environment. So really our first step is just a campaign to go sample and start sequencing who is actually there, so that we can start understanding what are the metabolites, what are the genes that are turned on that promote the particular pathogens in a P-trap in a hospital, for instance, to be able to propagate. And so we have to start at ground level, and it's really difficult because individuals think of the medical world and where we do have that understanding, and we're nowhere near that. I think we're, decades behind. So thank you.
All right, thank you for all the questions. We're running a little bit slim on time, so we're going to move on to the next portion for our breakout. So again, I'm Mark Sigrist. I'm with Energetics. You guys have heard me talk already. So we have some general questions to get started that you see on the slide. The first one regarding this topic. Regarding engineering approaches to mitigate disease transmission, what aspects of the underlying science are well known? So I'll give you a moment to think about that. And if you have comments, just grab a microphone, or if you want to raise your hand online, go ahead and we'll get started. So what is the science covering well right now?
I think from the air transmission perspective, the filtration, ventilation, and UV are more or less well known, have been studied, a lot of publications published on those.
Mm-hmm. All right. Is there agreement on the air measurements and transmission?
I'm just going to add, Nicole Rocky, Duke University. I am just going to add a slight perspective to that, and I think we'll talk about it later today. I think behaviors of individuals in settings have a large impact on whether things like ventilation or filtration will work. So while in theory, they are reducing infectious loads in an environment, I think understanding whether someone's talking to someone face-to-face 2 inches apart is going to be different than long-range transmission. And I don't think we know a lot about these differences in modes and their importance in different contexts.
Okay, thank you for that clarification. Claudia.
I mean, I don't know if this falls under engineering approaches, but vaccination.
Sure, yes, it's related, definitely.
I think from an engineering perspective, we know a lot about how to kill pathogens, right? Like from CT values and things like that. We just maybe don't know how to take into consideration the nuances of the microbiome and you know, not kill a mosquito with a chair and be a little more targeted.
Any other thoughts on what the science is doing well currently?
Thank you. I do a lot of work in low and middle income countries. So I always try and say, like, we've really improved our outdoor air quality, which has reduced our load of just acute respiratory infections, which worldwide are still the leading cause of children under 5. If you make it past the neonatal age, it's ARIs is number 1, and that's from indoor cooking with indoor charcoal, biofuels. It's outdoor air pollution from industry, and those point sources we've really dealt with. We know how to deal with, and we also know how to, you know, get away from cooking with indoor fuels, which in low and middle income emergencies is still the largest cause of child death. So I think there's a lot of places compared to 1950. We're improved. Right? There's a lot of work left to do.
Thank you. All right. We feel good about having discussed that a little bit. Get started. Let's move on to another question. So what scientific aspects of engineering approaches to mitigate disease transmission require further exploration or strengthening? I'm sure there are plenty of ideas. So who wants to go first?
CY Wu from University of Miami. And I think even though I mentioned about, you know, the ventilation, you know, the filtration, the mechanism of each device are well known, but there are social behaviors, but also the micros behavior, because they are part of the complicated matrix of materials encapsulating them. So how do they transport? And then how do these materials protect them from, say, UV inactivation? These still require more understanding.
Yes, and that goes to what the presenter earlier today talked about, the dust protecting the virus as it travels across oceans. Any other comments?
I'll just feed off of the comment that Carrie made earlier. I mean, we really don't understand how particular microbes function in the complex environment. So that's an area that we need to understand. And then the second one, which ties, is thinking about who we're designing for. You know, that goes to, you know, the middle, lower resourced countries, but then also, do we design for the child, the immunocompromised, the elderly, the average individual, and all of that is going to affect. I mean, we know that many of us are carriers of pathogens, but we are not sick right now that we know of. And so understanding that piece is really important as we're thinking about this.
Okay. Yeah, so since I think most of us are like experts in water or in air side, but I want to mention one thing is like the possible format transmission, the approach to control like the format transmission, because like I'm a mom of like a 1-year-old baby, so I'm always concerning about like the disease transmission in like the daycare settings. Lots of those like viruses or bacteria transmit through like the surfaces, but there are no very good approaches to control them, especially in the settings like daycare.
Thank you. Yeah, this is a theme that's coming through the day, is the lack of study and understanding about the aerosolization and how to quantify all of that in a big enough scale to help the most people. Any other comments?
All right, Andrea Silverman, NYU. I I mentioned this before, I just want to make sure it's logged here as well, is that we do know a lot about disinfection, but we don't know how pathogens are behaving, are situated in the environment in terms of sorption, aggregation, in vesicles, if they're have different structures that make them more resistant to disinfectants or not. So I think there's a lot more work to be done to understand that and to see if what we think we know about these organisms actually is there, works for them. I think another thing is figuring out what certain risks are. So antibiotic resistance, for example, and antibiotic resistance genes, there's a lot of concern about them. There should be, we think about, but what is the concern? Is it antibiotic resistance genes in a bacteria that's functional, or is it a free floating gene? What's the impact of DNA? Are there proteins that are of concern? So I think that there's like kind of understanding also, like, are we assessing all of the health risks appropriately? And so that are we actually targeting the right things? And are there certain risks in the environment that we should be studying more in terms of how engineered mitigation approaches work with them?
Yeah, thank you for that. I see a lot of nodding heads. Go ahead.
I'm going to just follow up.
Hold on.
Yeah.
Andrea, you brought something that I wanted to follow up on. We think of disinfection byproduct from chemistry perspective, like natural organic matter. Can you think of disinfection byproduct when you're tackling viral and microbial disinfection? What would that look like?
So the question is, should we be thinking about disinfection byproducts? Yeah.
Biological, as opposed to just natural organic. I mean, just for all of you.
Like the question is, are there potential byproducts of disinfection? So thinking like, could we be enhancing resistance to disinfection? I don't know. This is a, does anyone else have a thought on this? I should ask. Do you want to think about that for a second?
I guess I just wanted to bring up that we don't totally understand trade-offs like that. So when we disinfect for one thing, do we produce a byproduct that then is, toxic to some other aspects of the microbiome, or shifts it in a way that we did not intend that's harmful to human health or environmental health? So maybe just understanding some of those trade-offs a little bit better, and also at multiple scales. So like, we know that things at the small scale don't necessarily scale up. And when we have to do a model, sometimes we're tempted to take that small scale thing and extrapolate beyond the sphere of applicability.
And I'll just one more thing, that thinking about it is enrichment of certain organisms. So I know there's a lot of concerns about Legionella and other biofilm associated microorganisms. And if we're chlorinating, are we potentially increasing the concentrations of these organisms? So I would say that's an open research question too, is what are the downsides to applying some of the disinfectants that we are?
That's it. I think that's a great question. And I was at a Gordon conference and I just pulled it up. It's Lutgard Raskin from University of Michigan who presented on the impact of disinfection on the microbiome of the drinking water system. And it was really fascinating work that was exactly this, like thinking about what are disinfection byproducts from a biological perspective on the drinking water systems long-term in the biofilms and the microbiome that then is creating these kind of resistant organisms. So I completely agree. And I also just wanted to echo this. To me right now, like I have done a lot of work in water, and we actually have done some major studies over the past 25 years to identify like what are the actual organisms, viruses, bacteria, protozoa. We're not talking about protozoa here very much, but they do play a role that actually lead to the causes of death worldwide. And so we know like now the top 5, and we can name the studies, the GEM study, the other studies that give us that. So we know if we target rotavirus, it's the most reduction into childhood death, which is why you then do that. I feel like with the aerosol stuff, we don't yet know where the place to interject now is that will reduce the most health impact. And that's kind of, water was there 20 years ago. We didn't know what caused it. But there's been some key studies that got us there, and that's what I feel like is missing here. Like, should we be talking about cholera, Ebola, SARS-CoV-2? I think we can answer that, but I think we can't quite answer it in a non-pandemic context. So the risk assessment stuff really comes in, because it's all interesting, but what's going to actually have the most impact? So thanks.
Yeah, I think that gets to at least one of our additional questions for this session, talking about challenges as well as what would be potential next steps. Any other ideas on strengthening?
I just wanted to add that we also need to think about the fact that there's many microbes that are good. And, our tendency is immediately to think about those pathogens. And in fact, there's many microbes out there we know are associated with health. And so we need to be really careful about how we formulate that, that particular question. And I think there's a lot of work. There's been a lot of work thinking about how we make our buildings more energy efficient, for instance, and yet we don't really understand what that impact is then in terms of microbiome. And so this goes back to the operation of those built environments. And so the disinfection byproducts, thinking about that more broadly, I think is a really important question.
It's coming back to that, what might be protective, because that's actually what some of what we found in the studies is organisms we thought caused disease in the actual mix in the environment, when it's all together, actually became protective against other things. And that, what's protective, what's a risk?
Yeah, and we can think about all the children that had not been exposed to all of those particular disease agents in daycares, and then suddenly that huge spike in RSV that we saw. And so there's, all of that needs to be considered together.
Thank you. A lot of what we've just been talking about ties into some of the things that may have been ignored in the past research that relates to this overall topic. So we've been talking about that as well. Not to give this too short of a dedication to it, but are there any other things that you think have been ignored in research? Go ahead.
Good. So my name is PreMiEr. I'm from University of Nevada, Reno. So my past research have been focused a lot on like UV disinfection in water, in air, and also on surfaces. I feel that in the past 3 years, I have been working a lot with public health officials, doctors, and also just industry partners. I feel that one thing the scientific community have been ignoring or kind of things that we really need to demonstrate our result in a way that all the other parties, the public health officials, decision makers can understand. One question I keep getting is that, we are really good at generating result in terms of demonstrating, for example, disinfection. We know we can kill this bug using UV or disinfection chlorine at 99.9%, but how this translate into the in terms of the infection rate. Like, for example, we can kill the viruses in this room like 3 logs. Is that going to prevent us in terms of prevent us from infection? That's something we don't really quite know. At least I don't really quite know. Another thing would be, because right now we're talking about different solutions in terms of sanitization, disinfection, how we compare different disinfection approaches that actually requires a really standardized testing procedure and reporting, how we report our result, how we test the disinfection efficacy. And I feel that's something we are kind of missing right now. So yeah, that's just some of my thoughts.
I'll piggyback I'll piggyback off that and just say, in addition to disinfection and what doses will cause reductions in transmission, we also measure a lot of things, but we don't know what that constitutes in terms of reduction in transmission. So we're good at measuring pathogens in the environment now, not necessarily other microbes that might be beneficial, but yeah, we don't know what gene copies means in terms of, is that person going to get sick?
Carrie Hamilton from ASU. I think we have some modeling approaches to deal with these types of questions that maybe have been historically underused, like risk analysis, for example. And the second point is sort of unrelated, but kind of going off of something Andrea brought up earlier is, we're still using a lot of the same methods to study microbes where they are. And especially when it comes to biofilms, like, recently we were trying to get a representative sample of a submerged biofilm, and it's difficult to do. So, maybe some more method development on sort of in situ sampling to get those measurements of realistic communities could be helpful.
Works. Yeah, another comment follow up on Nicole and Carrie is that so far I don't think we have very good and quick method to like identify the variability, the real risk of like the pathogens in the environment. So we use qPCR to detect the gene copy numbers, or we use culturing methods to detect like only like the culturable portion of the pathogens, but we don't really have like a very good method to exercise the risk of like the pathogens that can really cause like the infections or the disease. So I think that is also one thing I think we can like improve in the future for like the quantification and identification of like the disease causing pathogens in environment.
Right. So you're saying it's not just the the viruses or the microbes themselves, but what they elicit and what could cause an infection. So you're able to detect the organism, but not necessarily what causes the result. Yes, variability. Thank you. Any other comments?
Yeah, if I may, just wanted to discuss the difference between water sampling and air sampling. So for water sampling, you can go to say EPA method, and this is how you do the water sampling and analysis. There's a standard method over there. Now, if you check the literatures in air sampling for SARS-CoV-2, you can see hundreds of methods being deployed over there, and there are no standard protocol. So there is no standard existing over there. I think that's the source of, say, you can say confusion or say incoherent of the data analysis and interpretation. So I think that's one thing we are missing in the air surveillance community because we don't have such a thing over there. So that needs to be developed.
Water sampling. For all environmental sampling, power and the time. But I think like standard methods. We need a lot better methods for all this stuff going on. For water, well as for air. Yeah, and I just want to echo that. Our colleagues that have the sort of like standards for like active based methods, but for environmental, like that's start. And I think I just also want to comment about understanding effectively and quantify method development even like are there ways that don't are and I think that's the point. The involvement of medical personnel. You think about it, infection, moves through the environment, gets to the body, and then medical side, and not even collaboration. And doing work internationally, and sciences and anthropologists and medical doctors and U.S. in particular have forced public health and engineering and medical. And I think something that I'd love to take from this, I don't know if we'll learn this physical engineering and public health and medical together in the U.S. And I don't think that we'll do that. And you may actually put a pullback further is, I think, the direction.
Yeah, that's a very good point. And that dovetails nicely into the next question is like, oh.
Great. I have not heard soil. So the built environment, soil there, I heard water, air, I have not heard soil. I would like to hear soil.
Fomites were mentioned.
Fomites.
Anybody here want to? I just want to remind everyone, yes, everything you're saying here is being recorded and documented by the note takers. It's recording in the Zoom meeting, so it's not getting lost.
Carrie Hamilton from ASU. I'll just mention that like in Arizona specifically, we have valley fever, which is a fungus that grows in soils, and there's not an understanding of where and why the hotspots in soils happen. So I think there's like this I don't know, environmental heterogeneity that maybe we don't understand for a lot of these different pathogens or contaminants.
Yes, soils we have to think about. We have to think about surfaces. We have to think about fomites. And we also thinking about what Carrie just brought up and some other things. We have to think about fungi, protozoa, and helminths also, especially if we want to have a global perspective. So I know a lot of us are talking about bacteria and viruses, and I think that's a lot what we work on, but making sure they're not forgotten and lost.
Yeah, and I'll just add to that as well. I mean, thinking back to when DDT was banned and thinking about how DDT was being brought in just from the outdoor into homes, and so we're very much thinking about it in that context. What are the how we're architecting our buildings that actually brings in and have those things stick around.
Thank you, and I think we've sort of evolved and moved into thinking about some of the opportunities. Some of them were long-term, like you said, moonshot, but some of them were more short-term, such as bringing various disciplines back together to talk about these things. Let's talk a little bit more about what are some of the short-term opportunities to help develop these mitigation strategies. Anybody have any other ideas? Go ahead.
I would say, for one, coming up with actual model systems that we can all work with. So not only the actual system itself and the methods, but what are the actual model organisms that we want to study that are representative of actual environments.
Thank you.
It was something that was brought up in the presentation earlier was the challenge with finding BSL-3 labs, or like even some labs don't have BSL-2 labs that can work with certain organisms. And so I think in terms of thinking about specific organisms that are risk, is there more partnerships with labs that can do high risk work with those that think more about engineering mitigation approaches, I think could be really like an easy short-term thing to do, yeah, good systematic studies, but looking at actual pathogens of risk.
Any other thoughts on short-term solutions or opportunities for short-term solutions?
Federal, like, okay, you are investigators funded by NSF. There are national labs that have these facilities. How do you see the interaction with national labs and others to augment the capabilities?
I think there's actually tremendous opportunity. I mean, feeding off of the comment that was just made, especially with NIST, I mean, they specialize in helping identify microbes that have similar features so that we can actually start answering those particular questions. So I think that's an area that we need to explore more.
Thank you.
One thing we've done is I have a BSL-2 lab, but not 3 or 4, but you can run a ton of samples in 1 and 2, and you can figure out the parameters that matter and run a thing of like different humidities, different temperatures, different this, and suddenly you have 800 samples. But then you can narrow that down and feed that to a BSL-4 lab. So like I work with a needle at BU. They don't care about any of the environmental stuff, but they care a lot about Ebola. And they're literally like, if you give me everything, we will run Ebola on it. So you run 800 samples in your lab and you figure out what really matters is plastic and wood, right? In an Ebola treatment unit. And then you give those samples to the BSL-4. And I think it's going to be this comes back to the partnerships. I'm never going to run a BSL-4 lab. But if environmental engineers work with those labs, and there's a bunch of BSL-3 and 4 labs, and we can narrow down the research questions, because for them to run 20 samples is $200,000 in months of time, but it's possible to do. And so I think, again, it's these partnerships across different genres that's really going to solve these problems. We're never going to solve these problems within engineering.
Yeah, I wonder if there's some sort of program where people can apply for like time. I think about like beamlines in like, synchrotrons and stuff where like, you have certain samples that need a special facility. Like if there was a way, maybe this already exists, I have no idea, but if there was a way to maybe apply, if there's a BSL-3 or BSL-4 lab that works with a certain organism and you have a question related, could you apply for time from those technicians in that space to maybe run some samples and get some important data? So that's maybe a model that could potentially work.
And a super funny story is these people run Ebola and BSL-4, which I think is crazy, right? And they literally couldn't do an iodometric titration kit to figure out the chlorine concentration, which I think is something high school students can do, right? And so you put those two things together and you get a lot of power, right? And that's where I see us needing to go.
It would also be great, to have some opportunities to send students and cross-train at the national labs. Those experiences are so valuable and, good for everyone involved.
Thank you, Director Diallo. You're talking about internships, correct? Yes. All right, thank you. Any other comments on near-term opportunities? All right, well, let's take a look at the next question. There was some discussion about this already, but what are some of the challenges you anticipate in attempting to pursue these opportunities? It can't just be funding.
I mean, I think one of the big challenges is identifying the particular disease or the particular endpoint that we're interested in. It's amazing when we think about how much progress we made because there was this global pandemic that everybody cared about, and there was huge amounts of advances and resources. But that's, in my opinion, this is the question I always get is, what is that endpoint that you want to study? Because we need that to be able to put our resources together to advance it.
Yeah, understood. Defining the scope is very difficult sometimes. What are the challenges to bringing different disciplines together? I know there are a bunch of those.
I was going to suggest that I think there's this false separation between the environmental media, soil, water, air, surfaces, and maybe by overcoming that a little bit, we can also make a better case for why environmental engineers need to play a role in pandemic prevention, not just from the medical side, but there's so much that can be done, from ventilation rates to disinfection to all these other things. We have a lot of tools that we're not fully leveraging.
I'll add one more to that. I think, feeding off of that, I think as academicians, we need to do a better job in our education system and how we adapt our environmental engineering curricula so that our students can actually start answering some of these issues. earlier than in grad school.
It kind of ties into what was said earlier about internships and working with national labs for training. Hopefully that can be a synergy. CY, you had a comment?
Yes, I wanted to actually bring another aspect. The pandemic, this is not the first time in human history, but do we really learn from the past history? And that's my question, because I remember in 2010, I was involved in Air Force Research Laboratory, and to prepare for a pandemic at the time it was the pig flu. And now I cannot find those documents. And it's just amazing how we handle the historical data. I think if these data are available to us widely, we know how to access them. I think that will shorten our learning curve in dealing with these problems. So the literature is really an issue, especially for those not in the archived journal articles.
Yeah, I imagine it's difficult to find older government-funded studies relating to pandemics and pandemic preparedness and response because of what happened at the beginning of this pandemic. Any other ideas on challenges? We can totally open it up. If there are other topics associated with what we've been discussing, feel free, go ahead.
I guess I'll just feed off of what you just said, and especially, you know, in the omics world, now that we have, I mean, the data that we're getting now are totally different than what we would have got in, you know, 2010 when that was happening. So now at least we have better data, but thinking about the FAIR principles of how these data are publicly available and how we actually can all use those. But then it goes back to what are the standard methods for collecting those and actually being able to analyze them. So that's something that our data will go much further if we can all agree on that.
I want to share one sort of idea of the research idea related to the sampling and then human behavior. Probably there'll be different solutions for different populations. For example, when you are talking about kids in daycare, the way they interact is very different from, say, adults sitting over here. The format transmission is probably more important for adults, but in this kind of adult interacting environment, probably that's not really critical. I mean, just combining the human behavior for different populations, and then the sampling technology will be an important topic to address in the future.
Yes, and we'll be discussing that after lunch today. So this afternoon we'll get deep into the human behavior side of things and risks associated with it. But thank you for the preview, CY.
Okay. So the sampling challenge. While down some challenges that
I think at least there are two important components over there that are the challenges. The first one is, like you said, the concentration is really low. So how do we concentrate the samples? You have to get a large air volume and then concentrate it into very tiny volume that you can process in for the analysis. And then second thing is, during that process, you needed to make sure that the viability of your microbes are not damaged. And that's even more challenging than the concentrating part. So these are the two things that we needed to work on in terms of getting viability information for risk assessment. But if your purpose is not to determine the viability, let's say a lot of wastewater surveillance, and it's the same if we do the air surveillance, the goal is to see the trend, not really getting down to proving the transmission, the risk. In that case, you needed to adopt different approaches. for different purposes.
I'll also add another challenge I think of when I think of air sampling is not knowing where to sample. In water, we often, we know the tap is where the water is coming into a home, or the shower is where water is coming in. People might be aerosolizing, breathing that in, but air is everywhere. And so understanding at what points to sample and what's going to be most important for understanding transmission risks is important there.
Another thing related to the difference between air and water sampling, I want to echo your point. So in water sampling, you probably go to the manhole to retrieve the samples, or where to, because water have to go through certain channels. But the air, for example, we were concerned about school or sampling in schools, or the kids there, are they safe or not, right? But even in the school, where exactly do you do the sampling? Do you do it in a cafeteria? Do you do it in a classroom? If you do it in a classroom, there are hundreds of classrooms over there. Do you go to everyone? And if you don't, like say, oh, everybody will go to the cafeteria, but is the cafeteria really a better location? So this is unknown that needs to be studied. And it's very challenging also because the concentration is very low. It's really a probability issue. When you don't know if there is a sick person that can discharge the pathogen, compared to, hey, I go to a hospital isolation room or isolation room in residence where we know there is such a person, such a source, the sampling strategy will be totally different.
Yeah, this ties back to the modeling and the issues, especially when you're dealing with noise and contaminants in these low biomass samples when you're trying to do the models. And that's what I alluded to in my talk, the issues that were uncovered in the fetal microbiome literature, where they thought they were focusing on the right organisms. And it turns out just because of how they were processing the data that was not accurate. So then it goes to what are the right controls to put into place for those types of studies. And I don't know that there's a good answer yet about what are the right controls for these types of studies.
I feel like one question I have maybe is, so when we think about water, we can sample water from like a stream or river because most people don't have, like 2 billion people don't have pipes, right? So we can sample centrally, and that's really hard because you have to filter a ton of it and you won't find it. And it's all the same thing you're talking about. If I'm trying to find something in a river, I just won't find it unless I sample hundreds of liters, and then it probably doesn't work anyway. But the human body is a concentrator, right? And so if you sample feces or wastewater, you have a concentrated sample, which is why it then works. So you sample feces and you grab what's in there, and then you sample wastewater. In air, there's 2 different things. There's the air in this room and the air I'm breathing out. Is the air I'm breathing out also a concentrator? And are we sampling the room, which is going to be very hard? Are we sampling infected people and the air you're breathing out? And that, I guess, is, I'm hearing some people talk about sampling air, but to me, sampling wastewater is an infected person's concentrated material, which is different than sampling water, which we have it's the same discussion. Like you try and filter a bazillion liters and you still don't get it, right? So and wastewater surveillance is about surveillance of infected people, right? So you have something to.
Just gonna add on. I was just gonna add on to that. So air sampling in infected people is also even hard. So sampling their air, you might not capture infectious particles, or you likely won't capture infectious particles. You might not even measure gene copies. When somebody's shedding in wastewater, it seems easier to detect, right?
Individual versus centralized.
Yeah.
So centralized is easy because you've got 4 million people if you have a wastewater system.
Right.
But individually, it's really hard. Like an individual sample is hard. So you aggregate samples. So that's an interesting, that's a distinction.
I think in the human body, we also need to consider that it's not only airborne, right? There's the gut, there's the skin, there's the oral, there's all of these different pieces that we need to consider. And so thinking about, the organisms that colonize in the built environment, what are the places, the body sites where they actually come in? And we don't we don't yet totally understand. Yeah. Yeah. So we need to, that's certainly a place that we need to model and understand.
So if I'm here, the interface between the microbiome and the built environment, can you guys comment more on that?
Are you asking in terms of what are the research gaps or, okay, yeah. Yeah, absolutely. I would say we really don't understand much and we don't have the sampling tools yet to be able to analyze that. So I'll say, we've done some work trying to test some like silicone bracelets, which are, passive samplers that are used for chemical exposures. And those are really difficult to use for human, microbial exposures, but we need lots of models that can really cross those, that can combine, I would say, the built environment and the human, and we're nowhere close to understand, which is why I think, what you mentioned, we go back to fecal samples because it's concentrated, it's easy. It's related to the gut, and usually, that's where in the human context, human health, that's the context we understand the best. But even there, we know that there's the gut-brain axis, and we know that there's many neurodegenerative diseases that are associated with brain function that are linked to the gut. But where do those microbes come from? There's lots of pieces. We don't, yeah, let alone where those microbes are coming from in the first place. I'll just say, for me, my biggest eye opener was a study that we did probably about 5 years ago, which was looking at children's guts microbiome, and we found organisms in these children's guts that were the organisms we use for bioremediation. So we found D. halococoides in these children's guts, which, wasn't something that I was expecting. But of course, it was, when you think about it, it's the way for those children's guts to detoxify from exposure to all of these halogens that are all around us. And so thinking about that as a sensor of what exposure is.
I have a follow-up question about what you've just been discussing. Is like, is there a possibility of, you were talking about the difficulty of air versus water and sort of surveillance. What is there a way to combine them so you get more effective air surveillance by using the water surveillance first? So you can target areas where you're looking for something specific and then do further studies about the air in those environments or buildings, or this is the layperson's question.
I think it depends what you're looking for, but maybe for the pathogens or microbes especially, we can understand better about how they're shed. So like, for example, human adenovirus 4041 not shed in respiratory secretions, but other adenoviruses are. So then you might not monitor in the air for that. You would monitor in the wastewater. So if you have a better understanding of that partitioning, I guess, of the emissions, then you can tailor your sampling approach accordingly and then map that to how we model things and how we address, the intervention. So maybe for COVID, more efficient for an air intervention than a surface, since that's the mode of transmission, but for something else, maybe that's not the case.
So in my postdoc study in Michigan, we are actually looking at like how the bacterial pathogens transferred from shower water to indoor air, and we try to like link like those abundance in water and in air. So in that case, like maybe we can use like those concentrated concentration in water to predict the release of pathogens to air. And we were actually also trying to work together to establish like a risk assessment for like this type of transmission, and hopefully like we will get more information on that later. Yeah.
Or can be like I see these putting water and all that. What happens if the soil actually?
Our presentation this morning talked just about dust. I mean, dust comes from soil.
Well, I'll just say from the antibiotic resistance perspective, like a lot of what we know is from the soil microbiologists studying like ARGs in soil. So it's clearly a reservoir. And I think the question is when it becomes meaningful to human health. And maybe we don't have a handle on that question, but I mean, you know, soil is certainly an incredibly high density of microorganisms, and it could be a sentinel like wastewater.
To come back to what people have talking about before with helminths, and I know Amy Pickering's done research in soil in homes with dirt floors, like we see a lot. And so this comes back to the built environment, like, do you have a floor or are you on a dirt floor? And Amy Pickering's research is showing living in a home with a dirt soil floor is really a risk. She's seeing a lot of bacteria and anti-resistant bacteria transferred via feet, on the dirt floor. But this is a built environment. Like we don't have dirt floors in Washington, DC. So it's less of a risk factor here, right? And so I don't know. And so what do we bring in? Like someone talked about bringing dust in. What are we bringing in? How? Yeah. And so what are we bringing in and what do we get when we're outside versus those numbers this morning of like, if we're talking about the built environment, how many hours do we spend on our pillow? Right. And so that's I think it's something we think a lot, especially with helminths.
Definitely something we need to think about especially we have more flooding, we're flooding animal waste, human waste, and it's like distributed across the land and then settles in. So what is the persistence of these organisms? Super important. I think that thinking about, and all classes of potential pathogens. I think one of the challenges in thinking about soil is the methodology. So with soil, it becomes even more, to talk about the challenges with sampling a water body and air, soil is even more challenging because it's not mixed. There's no mixing, right? There's like no opportunity for mixing. You have to really figure out where to collect your sample when it comes to soil. So experimental design is really important. I think also there's these challenges with actual like, we tend to know how to analyze liquids, right? Liquids, including air, fluids, I should say, not liquids, fluids. But with particulates, you have a new challenge where you have more potential for aggregation. You might need to extract off of a soil particle. And so I think that method development might be like, so thinking about challenges for soil related work, methods, and there's lots of methods out there. I just know that I'm intimidated personally by this is recorded off the record, but like, it's just, you know, like when you're so used to working with something that is well mixed and where you don't have to extract something off of a solid that might interfere with your results, then you need to kind of really think about your experimental design. So yes, but we definitely need more work figuring out how to do it well. Yeah, and lots of inhibitors for if you're doing any molecular work, for sure.
Yeah, I'll just add, I mean, we did some work in my lab looking at the different types of soil and them acting as reservoirs for antibiotic resistance. And we know that, for instance, if you have a clay soil, that antibiotic resistance gene is going to stick around for a really long time. And then, you know, those can then be transformed and, you know, conjugate into particular microbes. So I think what we can learn from it, though, is I think there are modeling frameworks to start thinking about from a chemical characterization and surface characterization. How can we start thinking about from a mass balance perspective where different organisms may live when we're bringing them in from the outside and thinking about it from that way. And then this goes back to the architecture and the operation of our systems, thinking about what are features that are more or less likely to allow the propagation of particular organisms that we're bringing in, or are we able to capture them through our HVAC systems, for instance?
I just want to build on something here, which is one of the things that I'm bumping up against is like, we can do a bunch of Ebola research with surrogates in the lab, with disinfectants in the lab, and then we can take them to an Ebola BSL-4 facility and we can look there. And then we can take that to World Health and say, how can we make policy? And they're like, we want data from Ebola treatment units. And I'm like, okay, we can even do that. We can get people into Ebola treatment units. We can take swab samples. We can answer some questions about how to maintain disinfection and prevent transmission in an Ebola treatment unit. And then we want to take a swab sample to a lab in Uganda and do infectivity, because that's what you need in the end to make policy. You've done all this work and like you actually need infectivity. And then they say, oh, but that swab sample is contaminated with blood or with dirt or with anything else. And we don't think our methods work. We don't think we can't get to infectivity because all of these methods don't work with contaminated or dirty samples. And I see that as a huge like next step of how do we actually deal with dirty samples in infectivity research? Are you seeing that limitation? Yeah.
Well, I was going to say something similar. So I think that there's a big question and challenge about taking the work that we do and then getting it into use. Like how do what we learn in the lab, how does it actually get used and believed? And sort of three thoughts. And one is jumping off what Danielle just said is that like, if we can talk more to those who we need to convince to find out what evidence they need, that would be really helpful. And so maybe workshops between practitioners and the lab-based researchers to understand what will they not accept, what do they accept, what is the gold standard for them actually, like what information do they actually need to implement the work that they're doing into the work they're doing. Because I think we all want to we do this work because we want that work to be used. And I struggle a lot personally about how do you make that happen. I think another thing is taking things to the lab scale to full scale systems. So what Danielle said, but also the interesting, like I was excited that you have this house test bed. So thinking about the built environment, HVAC and then premise plumbing and things like that, it's not easy to get the test beds that you can kind of control, but still make it uncontrolled enough to make it kind of realistic. And so having more opportunities to probably do, especially premise plumbing work in places that are actually used by people is helpful. And then on a totally different scale, just to add as final note, is thinking about antibiotic resistance. I think there's still this huge question about the importance of the environmental route. And I think that, yes, if you have a living bacterium that has an antibiotic resistance gene, that there's a potential for exposure, yes, that is definitely a risk. But what about what about DNA inside a dead bacterium? What about free floating DNA? And I think that's a big open question. And it's really, really hard to know what's the opportunities for horizontal gene transfer and incorporation of new genes. And I think that that's another thing that is a challenge in terms of figuring out risks and control strategies.
I'll add to that and say that it's also for environmental settings difficult to calibrate models of infection and risk, especially at a larger scale than, if you were a mechanical engineer, you would validate or calibrate your experiment by, running something over and over again, very specific in your lab, but we don't have that luxury of doing that. We have to look for natural experiments and look to kind of match up data from different sources. So that can be difficult.
The opportunity for using machine learning, data science, because you're not going to be able to understand all microbiome. Right? At some point, you're going to have some decision about how you're going to offer it to anybody in terms of how do you think about modeling this?
I think there's lots of opportunity there, and I think you're right. Like that we need more new data analysis tools, including potentially machine learning. I think one of the challenges that at least I know I face is I wasn't trained in any of those. And so sometimes it's hard to think about the first step. So, finding partners that are excited and willing to cross-pollinate in terms of both of us learning a totally new field to us. And so I think more opportunities for finding those partnerships would be helpful. Because yeah.
Turn green. Yes, absolutely. So we are doing work in that particular space, starting to think about how we can mine large data sets that are available. I will say the biggest issue for us to make advances in this space is we just don't have enough data sets that are with all of the omics technologies. So we don't have enough multi-omics sets. And so that's what we're particularly excited about, trying to bring to the community in these controlled environments, in our test beds, bringing those data so that we can start making advances in that particular space. But at least what we're starting with is really mining existing data sets so that we can develop the machine learning and AI infrastructure and then see if they actually hold true for the work that we do. We also have some folks working on metabolomics side, so starting to screen those to look for signatures of particular microbiomes and particular microbes in it. But I think there's huge potential in that particular space. The other side, though, that I'll mention, and this is going back to some of the societal impacts, we start getting into some tricky territory once we start looking at data privacy and start thinking about how we can start manipulating based off of these machine learning and AI methods. And so I think that's another piece of our work that we can't forget about.
We will certainly be talking about that more later today.
So another thing I want to build on that is that I think when we think about like data science, machine learning, especially when we deal with really complicated environmental sample data, I think data collection, data reporting is also very important, especially when we try to use the data from previous study, apply into a machine learning algorithm, then you can find that if different studies use different reporting techniques and different data collection methods, you may actually mess up your machine learning algorithm a lot, actually, because the experiment method, the reporting method are different. So I think there's certainly a need, depending on the specific research there's certain need to have a really standardized method for data reporting, data collection, especially in microbiome multi-omic studies. We need to really have a robust and comparable method for reporting collection so that we can really use data from different groups, different studies to really apply into this machine learning algorithm.
And I was just going to add that as well. We really need to combine not only the multi-omics and the machine learning in those, but also mixing them in with our physics-based models, which that is something, back to the comments we were talking about earlier, we do understand how things flow and we've got a much better understanding. That's what environmental engineers do. So really crossing to do this convergent science across all disciplines to be able to marry both the machine learning AI infrastructures with the physics models that we do. And that's really how we're going to advance it.
Yeah, that was exactly what I was going to point out, is that you don't have to choose one or the other. There's, hybrid mechanistic machine learning models. So I think just understanding which are appropriate to use for environmental modeling situations, and then also kind of understanding the implications of different choices, because we're not trying to sell somebody a web app, we're trying to prevent a pandemic. So I think there's a higher bar of what we accept in terms of error, right, in some of these situations.
I think an important aspect of that is translating it from what this group understands in terms of data research and results. Translating that for policymakers is a huge challenge, I'm sure. And I imagine a lot of you have had frustrations with that. So a lot of times I've heard numerous things that sound like, okay, that's a NIST job, but maybe academia can do it faster. Or maybe there's another way other than saying, oh, NIST needs to make a standard. So just a comment that I had.
I want to hear about training. Where would you change really thanks to the work with air?
think students need to become a lot more proficient computationally to exist in a modern workplace, like, learning AI, machine learning tools, learning some of these more advanced techniques that, maybe historically haven't been part of the curriculum.
Yeah, and I'll just add, I think we also need to think about what are the things that we need to take out of our curriculum, because we keep adding, but we're also losing a lot of students. I hear a lot of students come to me and they say, well, I can solve these problems, but I'm better trained in XYZ discipline rather than environmental engineering. And that's just a dagger to my heart, thinking about, we really need to think as a community how we can best solve this problem and how environmental engineers can really step up to work on these particular challenges. So there's a lot of potential, and I think lots of potential excitement to recruit the next generation of students.
I know I see a lot of science. Have an impact. So how is it that the environment organize some boot camp AI? I mean, that's thinking out loud. So why don't you do that?
Yeah, what are the barriers to being more proactive with training and developing, like what Director Diallo said?
I think there's probably some space for health and environmental engineering. Like I teach a public health engineering class that goes through the history of air and water and food and ends with obesity and gun violence and infectious disease, which are our challenges today, climate change. I think we do need to take some stuff out. We absolutely need to take some old classes out. And I think this keeps coming back to it, but I don't think one discipline is going to solve all these issues. I think we have to teach people how to work cross-discipline and do transdisciplinary training. And I think that's how can your piece fit into everything else. And I think too, there's something here. I've heard a couple different things here. Like, is the goal of this to prevent a pandemic? Because then we should be talking about stopping spillover, right? How do pandemics spill over into the human population? Or is the goal to prevent transmission, which is a different thing? Or is the goal to actually come back to like some of this? I imagine we're going to find out a lot of the diseases actually have a root microbiological cause that we don't know right now, right? Which is kind of what you were alluding to, right? And so like, are we about like finding out what chronic diseases are caused by? Are we about preventing transmission once a pandemic spills over? Or are we talking about preventing spillover of pandemic, which is a whole different thing? And I'm not sure that's been talked about as much here. So there's a couple places I feel like it could intervene, but it all is transdisciplinary. And I feel like I feel like the students, like they take hydraulics and they take this and they take this. And I'm like, oh my God, we've got to teach like how to write and how to talk to people and how to do what public health is and what machine learning. And your grad school is going to be 10 machine learning classes and your undergrad's going to be 1, right? Whatever. But there's a revamp needed. Yeah.
This isn't well formulated, but I think there's also a question about training at what level. And so like a lot of our undergraduates are going to go out into the world and be hopefully making an impact in the world and thinking about where students finally hear a bit more about pathogens and disinfection and microbiology often isn't till they're more specialized. So you may not have as much detail at the undergraduate level. I know that, we cover disinfection like this much, like so little in the undergraduate curriculum, and it's not until grad school, but are our undergrads then going out and making important decisions in terms of, either at the national or local level in terms of protecting people? And I think one of the challenges with environmental engineering is we are really diverse and we need to have an interdisciplinary understanding to really be able to do holistic work in this field. I think Mamadou, you guys have said that like the NSF program in environmental engineering is one of the biggest ones, and that, you read an environmental engineering proposal and they are all over the place because we do such diverse amount of work. And I think there's this interesting question of, does every student need to know everything, or do we need to start having sub-disciplines in environmental engineering earlier, thinking about the chemists versus the microbiologists and those? And so yeah, I think there's some really interesting work that we need to think about what needs every student needs to have versus when do they specialize. And even in grad school at master's degree programs, they're still very like really broad. There's more advanced work in every class, but it is still quite broad. So you can't get into some of the more like technically detailed methodologies and work.
Yeah, and I'll just add to that. I mean, I teach our environmental microbiology course, and most of the students in it are grad students, and they haven't seen biology since high school. So, and that, you know, when we're as environmental engineers, we're trying, I mean, this is what we do. We protect public health, and yet our students have, I mean, they don't receive that type of training. So I think your idea of, you know, should there be, you know, parallel tracks. I mean, I think of, you know, our biomedical engineering, you know, areas, they go in, one is more sensors, one is more, you know, human devices, or, you know, so I think we can think about it in that particular way. And of course, there's advantages and disadvantages of, you know, we'd then be asking an 18-year-old to be deciding what area they want to go into. And we know there's challenges with that as well. So I'm not sure what the best solution is, but certainly we can't have the status quo.
One other thought, I have some friends who did PhDs in microbiology. And I remember like doing practice defenses with them, like back when I was finishing up my PhD, and they were like, I didn't know that I could have done something like this, like work in a microbiological realm, but thinking about control strategies. So I also wonder if there's a way to bring more of this sort of control, sort of some environmental engineering principles into the realm of those that are microbiologists, because they, you know, like we talked about that, you know, our students have not taken microbiology or microbiology since high school or like the first year of college, versus students who have a full training in it, their whole, you know, academic training. And can we get more of them involved in the type of work that we're doing?
And I'll just add to that. I mean, I think this goes back to even what you said, just jogged my memory in terms of thinking about just transdisciplinary training at the graduate level, and the way most universities don't allow shared chapters as part of their dissertation theses. And so, you know, even thinking about how our graduate programs and how those are set up. And so I think it's even a bigger question at, you know, the academy level about how the barriers that we have put in place that, you know, are promoting a system that maybe isn't the best to actually answer the challenges that we have.
There's a low-hanging fruit called graduate student rotation, right? So you send a student to different labs with different PI that focus on, you know, a bio-focused environmental engineer. What, I mean, can you make use of that?
Students funded. It depends on the student. There's a bunch of programs I know that have started. They have, there's some type of, there's a bunch of schools.
And I'll just maybe ask a question slash, when I've seen rotations happen, it's usually within a specific department. It's not necessarily cross departments. And so that would not help what we're talking about, but if there were institutes or programs that crossed many different departments and allowed people to rotate through those different departments, then yeah, that would be wonderful.
You don't have to come to the environment. Program director. You just have to say, yes, I want to do these, then
Last comment, Claudia.
I'll just add to that. So our ERC actually came from an NRT. We had one of these national research traineeships, but one of the big disadvantages of this NSF model is that it's 5 years and it's done. The NIH equivalent, the T32 program, is one that you can be applying in perpetuity. And so to be able to meaningfully set up these convergent science education programs that are transdisciplinary to allow the rotations, there has to be another mechanism to be able to sustain that in the long term. So if we can extend the NRTs, that would be awesome. I'll just put my plug there.
And I just want to echo that because we had an IGERT that let us do a lot of this, and the IGERT ran out and it became the NRT. And a lot of the work that was allowed us to do stopped after 5 years. The 5 years was great, but I would just want to echo like exactly that.
So it sounds like there's going to be some follow-up with you at NSF. Indeed. All right, any more last comments before we break for lunch? Thank you, this has been a great discussion. Go ahead.
I'm just going to add that sometimes aspects of environmental engineering are a tough sell for students, like processing wastewater for 5 hours in a day. So if there's ways that we can make it so the student can see impact faster, I think that will entice them to stay. Is that some of the
Thank you, it's a good point. All right, we're going to break for lunch.